Welcome back. The session is today we will talk about gel electrophoresis. Two lectures divided into an hour or so after the break. And I'm very glad to introduce to you all Dr. Vasanth Bhaskara, my colleague from Department of Chemistry and Biochemistry, Ramaya College of Arts and Sciences. Dr. Vasanth has pursued his PhD from Central University, Hyderabad in cancer biology. And further, he has motivated and pursued very hardworking postdoctoral experiences in US in two prestigious institutions. After that, he joined our institute, and since 5 years, he's working for the betterment of the department in terms of academics and research activities. To his bench, he has a lot of research articles published in cancer biology. One of the compound resveratrol he was working. He published several prestigious reviews and articles. Welcome, Vasanth. Welcome to the QCA batch 2. And he will talk about gel electrophoresis. But he might ask some basic questions to test the knowledge of the students where they are. Maybe you are from diverse background in chemistry. After that, based on that, he will take the pace of the lecture. Welcome, Vasanth.
Thank you. So I can sit and.
Yeah, you can follow.
Yeah, I can
continue.
Very good morning, everyone. First of all, I would like to know if my voice is clear to everyone and you are able to see the screen.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay, thank you. So today, my topic is going to be on gel electrophoresis. And I know that many of you are from chemistry background also. So please do not hesitate to stop me anywhere at any point of time if clarification is required or if you have any questions. Okay, so don't hesitate for that. Right? Shall we start the session now?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
So I'm going to give you the summary of all the different electrophoresis techniques today. And it may be a little bit in detailed version of this for you. So you have to have a notepad and please note the flow of these contents. Okay. Right?
Yes, sir.
Okay, sir. Okay, sir.
Yes, sir.
Yes, sir.
So to start with the electrophoresis topic, let us start with the historical perspectives of gel electrophoresis. And as you can see on the screen, it was in 1937 by Arne Teslius. You have his photograph also on right side. And he was known as the father of electrophoresis. And he was a pioneer of the primitive version of the electrophoresis that is known as moving boundary electrophoresis or free flow electrophoresis apparatus. So that was very primitive and early times, and they used the separation technique in solutions. And the U-tube apparatus, that is the moving boundary apparatus that they have used, one of the arm is acting as an anode and another arm is acting as a cathode. Now your sample, which is supposed to be separated, is in the solution. And when electrophoresis is carried out, the molecules, particularly like charged molecules, mainly it is applicable for the separation, and that will move to the respective electrode, and that the way isolation of that particular compounds were done. So basically electrophoresis means phoresis is separation of biomolecules or charged molecules in the presence of electric field. Okay, so that was the primitive idea, and it came as moving boundary technique, and pioneer was Arne Teslius. Then the next version of electrophoresis is zone electrophoresis. So in 1940s and 50s, and pioneered by Oliver Smithy, And this scientist, he separated the biomolecules on solid supports, and that is zone electrophoresis. So different types of solid supports like nitrocellulose paper, cellulose acetate films, agarose gels, polyacrylamide gels, glass beads, granular starch, polyvinyl particles. These are examples of the solid support that they used for separating the biomolecules by applying the electric field. Of all these, agarose gels and polyacrylamide gels, these are widely used for separating the molecules. You might be knowing, right? Is it clear?
Yes, sir.
Yes, sir.
This is the zone electrophoresis. And at present, the high-end version of the electrophoresis is nothing but capillary electrophoresis or capillary zone electrophoresis. This was pioneered by Stellan, and this is mainly separating the biomolecules in the capillaries. Capillaries, you all must be knowing, these are the small tubes which you may be using, particularly for TLC, spotting the samples, right? So such type of capillary tubes are used, and you have in the illustration, there are two anode and cathode chambers which are connected by the capillary tubes, which has got the samples which are supposed to be separated under the electric field. So this is the capillary electrophoresis, and the next part of today's lecture will be in detail about the capillary electrophoresis. Understood? So these are the three different era or versions of electrophoresis.
So one simple question, if any chemistry people are there, so Vasanth sir is talking about biomolecule separation, right? Can anyone tell me about the basic unit of nucleic acid or basic units of proteins? What are nucleic acid made up of? Proteins made up of?
Amino acids, sir. Amino acids, sir.
I will take Abhijit. Can you tell me the basic unit of proteins? Abhijit,
Amino acids, sir.
what is the charge for them? Do they have a charge?
Yes, sir.
Do amino acids have a net charge?
What are the functional groups?
Functional groups in an amino acid. What is a peptide bond?
COOH and NH2.
Very, very good. So the net charge you think of, excuse me, think about it.
Okay, sir.
Same way, can Elizabeth George, can you tell me about what is the fundamental unit or basic building block of nucleic acids?
Amino acids.
Nucleic acid. Asking for nucleic acid, not
Nucleotides.
Nucleotides.
okay. Good, good. So
Phosphate group.
yeah, phosphate group. Can you tell me the charge? Does it have a charge? Phosphate group.
Negative.
Negative.
Negative charge.
Yeah, P43 minus, right? I'm correct. So yeah, nucleic acids have negative charge. Amino acids positive, and depending on the net charge.
We are going to discuss all those in detail.
Okay.
As I said, please stop me if you have any confusion or clarity that you need further, because I know chemistry students are there, my students also there, Himani is there, and if you are not following, do not hesitate to stop.
Yeah.
If you ask, then only I can respond. I don't know
Whether
at
you
what
are
point you people are
missing
stuck, right?
Okay. So basically the biomolecule based on charge, negative will move to positive, positive will move to negative.
Yes,
That's it.
yes. Okay. So this charge separation is a basic principle of electrophoresis. As I said, phoresis is separation. Under the electric field, you are applying positive and negative electrodes, electric field, and then molecules which are charged, they are tend to move to respective electrodes. Then you can collect in the pure box. Understood? This is the definition as of now, and you are going to appreciate in next slide furthermore. Okay. So you understood as of now the three versions that we have discussed now. Different
Yes, sir.
Yes, sir.
Yes, sir.
different forms of electrophoresis. That is, it started as the first moving boundary or free electrophoresis. Then second versions, they started separating the biomolecules on the solid supports, and that is zone electrophoresis. And the improvised version at present. The high-end version is separation in capillary tubes, right? So now we are going to discuss further in continuation with our topic, that is principle. So what is the principle of electrophoresis? It's very simple. Separating the molecules under the electric field of particular electric field strength. And this electric field strength, how much voltage that you apply, that much electric field strength will be created. And you can see in the slide, the formula is there, the expression. So the velocity of a particular molecule in an electric field when it is applied depends on the net charge on the molecule and frictional coefficient. More the frictional coefficient, less will be velocity. It is inverse relation, right? And lesser is the frequency, more will be the velocity. And what are the physical factors that can impact on the molecule which is moving during electrophoresis? So these are the native charge, that is charge, native charge. What is the term native here? Native means the original state. Suppose for any protein, native protein refers to when it is in the physiological state, that is in the living system. For example, albumin is there. You may be knowing albumin. Albumin present in the blood. What is its structure? Okay, that is called the quaternary structure. Proteins can be primary structure, secondary structure, tertiary and quaternary structure. So in the quaternary structure, you have all the subunits if it's a multimeric protein, and that is the form under physiological conditions which is functional. Proteins like enzymes, if it is in native form, it will convert, it is able to act on the substrate, converting them into product, right? So that is the functionality of the enzyme or functionality of the protein. And that is retained in its native state when it is in the living system, in the cell or in the body, blood. And when you take out from the living system, even in the test tube also, you can maintain the activity. You know that? So if you provide the conditions required for, the ambient conditions required for maintaining the activity of the enzyme or any protein, that you can maintain the charge, original charge and original status, original conformation is the term we use, not conformation, it is conformation. Conformation means the native structure, the structure of the protein in the physiology. Okay, understood. So what is the charge of that particular protein? Different proteins are there, different proteins will be with a different charge, and this charge depends on, if you know, there are 20 standard amino acids, there are acetic amino acid and basic amino acids. Different proteins will be vary with respect to number of acidic and basic amino acids, and that decides what is going to be the net charge. How many acetic amino acids, how many basic amino acids in a protein, and that is going to decide what is the net charge of the protein. Understood? And that refers to the native charge and native status. Understood, all of you? So that is the native charge of a protein is going to have an impact. What is actually, whether it's minus, if it is more of the acidic amino acid and basic amino acid, then plus charge, along with mass, shape, temperature, and some other minor factors can also influence on the movement of this molecule, because the separation is mainly relies on how the fast or the velocity of these molecules under the electric field that are being separated. Understood? So another expression here you can see, for the molecules under particular electric field strength which you have got with the applied voltage is dependent on the distance between the two electrodes. You have the two electrodes in the electrophoresis, and you know the average electrophoresis, if you have done, if you have seen, you have smaller apparatus, bigger electrophoresis apparatus. In that small and big, there is a matter of distance between these two electrodes, right? And that distance, if it is more, then if you are applying particular electric field, you have to accordingly change if it is smaller, if it is bigger. So distance is inversely related to the electric field. Okay, and electric field is directly related to voltage. So if you apply more voltage, electric field will be more for a particular distance. Understood? Between the electrodes. So now here you can see in the illustrations, always negative. I keep telling to my students, negative, if you have seen, like chemistry students also, electrode, you know, negative thoughts, negative, black color. So black color, those who are liking, they are having negative thoughts. So this is just simply, I'm kidding, okay? So this is the way you have to remember this negative electrode and positive electrode here. And you know, cations, sodium cation, chloride anions, anions move towards anode, cations move towards cathode. So which electrodes are those? You know, right? Plus will never go to plus. So plus always will go to minus. So cation, sodium cation is a positive charge. It will move towards cathode. So what is the charge of cathode then?
Negative.
Negative.
Right. Understood. So now you should not forget this concept. I told black is negative, and then sodium cation, chloride anions moves towards the cathode and anode. Clear? So that way, the separation of these molecules under the particular electric field strength relies on the distance between the electrodes. Understood? And also, you can see in the illustration, there are different type of molecules. These are like rod-like molecules. These are spherical molecules. And then they are also different with respect to the charge also. Okay. And small molecules with a particular charge, if it is highly charged, then it will move faster. And if it is small molecules, it will easily run away and it can move faster. And bigger molecules, they are lazy stuff, they will be lagging behind all the time. Okay. So that is about the principle of electroforces. Any questions you have, I can take now. Anybody, any questions? Okay, if you don't have any questions, I understand for the moment that you have followed. And next topic we are going to discuss is what are the different types of electroforces. So electroforces types are different based on the purpose, format, and then the gel composition. Now, based on the purpose, different types of electroforces are like agarose gel electroforces. Chemistry students should appreciate this agarose gel electroforces you might not be knowing. This is used for DNA, and this is the oldest form of zone electroforces. Agarose is a solid support, right? That's why it is zone electroforces. Then native page electroforces. Native, as I said, now you know, native means the native structure of the protein, which is actually the quaternary structure of the protein in the living system. And then this is also called as non-denatured. If you denature the protein, then it is reduced or denatured, and that is called SDS page, which we use for the protein. SDS page is sodium dodecyl sulfate polyatomic gel electroforces. This is widely used for separating the proteins. SDS in front of page is the reagent which is used as a detergent that will denature the protein. So that's why it is called denatured and also called as reduced, because there is a reduction of the protein to peptides. And here, if you don't have SDS term, that is native page. Okay. Then other format which is also used in the proteomics laboratories, that is isoelectric foxing. This is also called as tube gel, and this is called two-dimensional gel electroforces. Then there are immune electroforces techniques and pulse field gel electroforces techniques are there. So these are the more or less widely used in different laboratories depending on the purpose. Then these electroforces can be either horizontal, it can be run as horizontal, like agarose gel electroforces, if you know, and flat bed, then tube gel electroforces. Okay. Then based on the gel composition, if the gel complete for example, agarose gel electroforces is same, but SDS gel, if you see, there are distinct regions within the gel. They may be different with respect to concentration of the polyacrylamide and with respect to the pH or pore size. So within the gel, they may differ with respect to different parameters, and in such case, that type of gels are known as gradient gels. There's a gradient, okay? And non-gradient means the gels are continuous, as I said, like agarose. And for DNA sequencing, one type of technique is used, that is by using polyacrylamide gels only, and that is again continuous. SDS page is for proteins, okay? And polyacrylamide for DNA is used only for sequencing. Otherwise, for DNA, most commonly used electrophoresis technique is agarose gel electrophoresis. Understood?
So
Sir.
purpose of immune electrophoresis and pulse field gel electrophoresis.
Yes. We are going in detail. Next topic is going to be in detail discussion of all these techniques. So I'll discuss. For the time being, classification, you should understand how the electrophoresis techniques can be classified based on the purpose, based on the format, and based on the gel composition. Somebody, if they are saying gradient, this is a gradient gel, then now you know what is gradient gel. That is, they are having different regions with respect to any of the concentration, pH or pore size. Okay. Is it clear now?
Yes, sir. Yes, sir.
Those techniques in this session, I'm going to discuss with you a bit in detail. We are starting with the oldest format of the gel electrophoresis, that is agarose gel electrophoresis. Agarose gel, as the term says, the solid support. This is a form of what type of technique it is with respect to the generations?
Zone electrophoresis.
Zone electro.
Clear to everyone, right? Anybody is having question, doubts, you please discuss with me now. I said that zone electrophoresis is one type of second generation technique where the solid supports are used for separating these molecules. So agarose is the old standard method which is used for DNA to separate. You are also going to appreciate why it is specifically for the DNA. Okay. So now agarose gel electrophoresis, if you see, and if you have seen already, good. Otherwise, you have to see somehow, sometime in your next. See, agarose, if you dissolve in the buffer, TAE buffer, then you are going to boil it about 65 degrees centigrade. Then if you cool it, it will form the gel. Okay. So this is the way we used to solidify the agarose gels. And then we separate by using the cone, we make the wells and we load the DNA samples. Then if you apply electric field in a tank buffer is having salt, so that way electric field will be applied and DNA gets separated. This is how we use. And this polymerization principle, if you see, this agarose monomers, basically, they have got intrahydrogen bonds. And upon boiling, it gets denatured. So when you boil it, it should become completely like watery material. Okay. And this watery material, when you cool it, the intra-hydrogen bonds are becoming inter-hydrogen bonds. And when it forms inter-hydrogen bonds, this is the way the meshwork of agarose is formed. And that is called polymerization of the gel. Understood now? So agarose gel polymerization is basically denaturation of intra or changing of intrahydrogen to interhydrogen bonds. That is what gel polymerization in case of agarose gels. Okay. So that's how now agarose gel polymerization is used for making the gel. And these agarose gels, as I said, particularly they are useful for DNA. So DNA you all know, right? And protein, if you see, and if you compare with DNA and protein, relatively proteins are spheroid molecules, spherical, and DNA is a lengthy, long, rod-like structure. Why it is used for DNA, particularly agarose gel electrophoresis? So why? Because there is a term known as axial ratio. What is axial ratio? Ratio of longitudinal axis to transverse axis. This is longitudinal axis and this is the transverse axis. If you see, the axial ratio is very high for DNA. If longitudinal axis is more, automatically it is the rod-like structure and a long structure. Particularly for the molecules with higher axial ratios, agarose gel electrophoresis is suitable. Not for the spherical molecules with a lesser axial ratio. Understood? That's how. This is a concept related to agarose gel electrophoresis. Why? For DNA particularly, it is applicable because of its higher axial ratio. Understood?
What is the need of taking axial ratio in gel electrophoresis?
We are not taking the axial ratio. This is the concept based on which the molecules which are appropriate for agarose gel electrophoresis. If you do proteins, they are spherical, small size molecules, you cannot get separation. Understood? The pore size. Okay. Agarose gels, you have enough pore size, and DNA is a bulk material compared to protein. And this concept is explained in terms of axial ratio. We are not taking axial ratio, but this is the concept for why DNA can be separated by agarose gels because of its higher axial ratio. Okay.
Yes, sir.
Now, as I said, you have polymerized the gel. You know the concept of what type of molecules are appropriate for running on the agarose gels. And when you run agarose gels, DNA we are using within the gel, we are using ethidium bromide and it's a fluorescent material. We are creating the wells. After boiling, when you denature, it is in liquid form, and when you keep it at room temperature, after some time, it gets solidified. When it gets cooled down, you will get the gel. And when you load the DNA, when you apply electrophoresis, then separation. This is marker here, ladder, and you have the different DNA samples. You can see how DNA electrophoresis gels looks like. Okay. Wherever fluorescence is there, that is the DNA which has got separated. And separation here at different positions. This is a very important point as far as analysis is concerned. This separation at different positions are with respect to their size. And size you can make out based on the marker. This marker, you know what is the size at which position of this fragment, what is the size, and you have to correlate with your sample the fragments, and that way you can make out what is the size. Okay, understood? So that is the way we use this technique for separating the DNA. Now, how this DNA is moving during the agarose gel electrophoresis. So there are two models were proposed how DNA while electrophoresis is moving across. This is biased reptation. Repta means something like reptile. Reptile is snake, like a snake-like movement. DNA is moving through these agarose gel pores. Another theory was proposed, that is caterpillar model. In this, DNA is forming like head and tail structures, and it will be getting extended, a kind of pulling. Okay, that is extension. And this, like caterpillar, in this way, it will move through the agarose gels. So this way, two models were proposed for DNA movement during agarose gel electrophoresis. That is all about agarose gel electrophoresis. So what are the applications when we use this agarose gel electrophoresis? As I said, mainly for DNA. DNA is charged material, and it is very good for electrophoresis technique to separate. To isolate genomic DNA, then to purify to PCR, that is used during cloning. So PCR amplicons for purification, for characterization, all these related techniques, agarose gel electrophoresis is the basic technique that we are using. Fine. So that is all about agarose gel electrophoresis. Again, I just wanted to know any questions, clarifications you require from this concept. Is everyone clear?
Yes, sir.
Yes, sir. Yes, sir.
Okay, good. So next, another technique is pulsed field gel electrophoresis. After agarose gel electrophoresis, I am telling this pulsed field gel electrophoresis because this technique also used for the DNA. What is the difference between agarose and pulsed field then? Pulsed field, this electrophoresis technique is used for bulk DNA material. Okay, so DNA when it is heavy material, particularly during taxonomy. Okay, when naming of plants, nomenclature, this technique is used, and when bulk DNA separation is very challenging. That is something like 50,000 kilo base pairs of DNA. When you are separating, this technique is using alternative forms of electric fields. For some time, in one direction, DNA gets separated, and then in second, following that, in the other electrodes will be turned on. Then again it turns off, and another direction, electrodes gets turned on. This way, alternative directions of electric field is responsible for separating the bulk DNA. This is the basis of pulsed field gel electrophoresis used in taxonomical studies. Okay. So that is all about the gel electrophoresis techniques, which is applicable for the DNA. And now for the proteins. Already I discussed enough about the native protein concept, and you know what is native protein means. The protein which is functional in its quaternary conformation, which is in the living system, the protein is completely functional, and that is said to be native conformation. So if you separate, or if you want to characterize, or if you want to analyze such type of native structure of the protein, and that is under non-reduced conditions. SDS is reduced condition. So here native means without SDS, that is non-reduced or non-denatured. Protein, if it's denatured, that is denatured. Non-reduced or non-denatured, it's in original state, in its functional state. As I said, from the living system, you can take out the enzymes in the test tube, and you can, or in the gels here, and you can maintain the condition so that it will exhibit the enzymatic activity, and it will convert the substrate into the products. Okay, that is the basis here. And this type of technique is called native gel electrophoresis. Native gel electrophoresis is used for analysis of the proteins which are in its quaternary conformation, which are functionally active. So these are called activity gels. This is one technique, and another one is gymogram. So activity gels, you see, for example, this is a protein here. LDH, lactate dehydrogenase, is a protein which you want to characterize. Okay. You know all right, enzymes are the proteins. Enzymes are biocatalysts. Chemistry students know that catalyst and its function in the reaction, correct? Same way in biology, we have biocatalyst, that is enzyme. It will convert substrate into product. Here LDH is one of the enzyme which will act on lactate, converts into pyruvate. In doing so, this is also responsible for reduction of NAD. This is reducing equivalent. NAD is getting reduced to NADH. Reduced compound NADH is here with hydrogen. And this reduced compound formation in turn responsible for the reduction of the formagen. Formagen is one compound. If you use this salt, tetrazoleum salt, okay, this will be converted to formagen insoluble crystal. And that is the principle of this activity gels. So now here you see your samples here, when you separate it, wherever the LDH, for example, in our example, in the illustration, so you have the bands, these are lactate dehydrogenase. And when you add the tetrazoleum salt, because of its enzymatic activity, as it is a native gel, as you are maintaining all the conditions required for its native structure to maintain, it will retain the function, and it will convert tetrazoleum to pharmagen. And this is how you see a pharmagen insoluble precipitate will form the band. That's how now you can see what are the different isomers of this lactate dehydrogenase. You can see that way. This technique is called activity gel, and which is a native gel electrophoresis technique. Then another technique which is used for native proteins, that is called gymogram. Okay. So this is for particularly a group of proteins in cancer biology. There are some proteins known as proteases. Protease means the enzyme which acts on the protein. It digests the proteins. Okay, these are one group of proteins. As I said, in cancers, you all know that even chemistry students also know cancer is basically a kind of abnormal growth, and their cell proliferation, cells will divide, right? That is apparent, means abnormal. When cells divide at a particular stage, the cells will become abnormal and it will become loose. Then cells will come out from the primary site, enter into the blood and lymphatic system. Then it will travel to the other parts where metastasis, that is called metastasis. All these cells will seed into distant parts of the body, and again it reforms the secondary tumors. Understood? And the cells becoming loose, I said, right? This loosening of the cells is because of the action of proteases. At this particular stage, these cancer cells will secrete proteases. It digests the extracellular matrix. Extracellular matrix is a fevistic, glue-like material which keeps the cells intact in our body. Firm, it will be firm. That is healthy status. But in cancers, because of these proteases, it digests the surrounding extracellular matrix. In that way, cells will become loose. Then cells will eventually enter into the blood and lymphatic system and can that way move to distant parts. Now understood, right? What is the protease and its role? So this is basically again protein. In such type of proteases to characterize, if you want to use some drugs, you have found now drug in Biocon and you want to test its effect on metastasis. Particularly if you see these proteases, then you can stop the metastasis of the cells, correct? Now, these proteases can be studied by the technique known as gymography. Okay, gymograms are the gels, and these proteases in cancer cells, I said, these are called matrix metalloproteases. There are so many MMPs are there. These are proteases. And as I said, these proteases are secreted by the cancer cells and they act on other proteins, that is extracellular matrix, a glue-like material that keeps the cells in the tissue form. It's a glue, fevistic, If you put fevistic, it will stick together. So cells are stuck and maintains its integrity in the tissues because of the fevistic, that is not fevistic in the body, but it is extracellular matrix, and that is a meshwork of proteins, and that is digested by these proteases in the cancer cells. So I said, now if you stop these proteases, then you can achieve your, you can stop the metastasis, and it's an important aim in drug discovery industries now. Okay, so this is the gymography, and what is the principle of this proteases to observe? As I said, these are proteases which will work on which will digest other proteases. So your gel, if you have seen, I said that most of you might have seen the gels. While making the gel polymerization, you add the protein gelatin. So these are called now gelatin gels. And these gelatin gels, because of the proteases in your samples, it will digest. There is a stain. Here also we used Kumasi, but here only the bands which are positive, they are blue color. But here the reverse. So the gel is blue color, and wherever the proteases are there, those are appearing lucent or unstained. Okay, why unstained? Because these are the regions where proteases are there from the sample, and they acted on gelatin which is present in the gel, and this gelatin is a protein. So this is protein is not there. There is no protein because of the protease, and when there is no protein, it will not be stained by Gumasi. But in the other parts where there is no protease, you have the Gumasi blue, and that's how you can make out. And this is very, very, very precise. Okay, wherever the small amount of the protease is there, you can see here how much precisely these bands can be analyzed. Okay, understood? So these are the two native gel electrophoresis techniques used for proteins in its native conformation when it is fully functional. One is activity gels we discussed, another one is gymogram gels. Any doubts, questions, clarifications? You please let me know.
Sir, what is the purpose of adding the chromogenic substrate in activity gels?
Gelatin, right?
Chromogenic substrates, the purpose of adding the chromogenic substrate.
No, chromogenic substrate. Yeah, chromogenic substrate in activity gel is nothing but here. See, this is tetrazoleum salt. This is the substrate now. This substrate is converted to chromogen. Chroma means color. Genic means generating. Color generating substrate. Here, what is the chromogenic substrate? Tetrazoleum salt. Because this is forming insoluble colored formagen crystals. Understood?
Sir, okay.
It's a good question. Good question. Okay. I have here. So that way you please make out, even if anything I'm not explaining, you have to remind me. So chromogenic substrate is a general word wherever, because of the reaction, if color formation occurs, that compound you can call it as, name it as chromogenic, and it is a substrate because it is acted upon by that particular enzyme. So if it is clear, the discussion of native gel electrophoresis, then we are going to move forward with the reduced or denatured or STS polyacrylamide gel electrophoresis, which is very widely used in proteomic studies. These are very basic techniques for characterization of the protein samples, STS polyacrylamide gel electrophoresis. Okay, so this SDS page technique, I'm going to explain you in terms of two broad headings, and you can see the concept can be explained as the first broad heading, the sample preparation. So two steps here, one is sample preparation, and another concept we are going to discuss about the gel polymerization. Okay, so first sample preparation. Sample preparation for the STS page is nothing but denaturation of your protein and binding of SDS. As a term, SDS page, the important component is in the sample buffer SDS. What is SDS? Sodium dodeca. Dodeca is 12. Lauryl is also known as lauryl. So SDS is sodium dodecyl sulfate, or it is also known as lauryl sulfate, 12 carbon. This is nothing but a soap, detergent. It's a soap, you know, right? Soap. So this is an anionic detergent, and detergent when you use, basically the lipid greasy material can be removed because it forms the micelic structures. So here proteins will be denatured because of the STS, and next we'll see how the role of STS is there. So this is the STS molecule. It's a long chain hydrocarbon and with a sodium sulfate salt. So this is nothing but a soap. So this is the sample buffer that it contains. We already discussed STS in the sample buffer. Then another important component is beta mercaptoethanol. So beta mercaptoethanol, this reagent, if you see the smell, you can never forget in your life. Okay, and even triazitol, DTT is another one. Don't forget. Okay, whenever you get an opportunity, please nicely smell this beta mercaptoethanol. If you have cold and all, immediately it will get to you. So don't smell. Okay, I'm just kidding. So this is such a pungent smell. This beta mercaptoethanol is converting cysteine. You know, 20 standard amino acids, sulfur containing amino acid is cysteine. So cysteine, two are there. Cysteine If two cysteines are connected by interdisulfide bond, that is called as cysteine. And if it is cleaved and cysteine with sulfidyl group, that is one cysteine is called as cysteine. Okay. If two cysteines are interconnected, that is called as cysteine. So these cysteines are acted upon by beta mercaptoethanol and gets reduced to irreversibly cysteine. What is this concept having role? See, this is the protein, for example, here. In the protein, you have cysteine bonds are very important, the cysteine bonds, which will make interdisulfide linkages and for the quaternary structure. Okay. What is the quaternary structure? This is one polypeptide. Quaternary structure, if you know hemoglobin, you have 2 alpha, 2 beta. All these monomers are connected to these disulfide bridges. That way, 4 quaternary monomers are connected and form the quaternary structure of hemoglobin. Okay, and that is a fully functional state of the hemoglobin quaternary structure. Now, for sample preparation, what you have to do, you have to make it individual alpha. You have to denature the protein. Okay, that is what we are doing here. This is a reduced or denatured gel electrophoresis. You are not worried like in native gel electrophoresis for its function or the nativity of the protein. So for this purpose, the interdisulfide bridges are very important for maintaining the quaternary structures. Intra for the particular structure, inter for making the quaternary structure, that is like 4 units in that alpha, 2 beta, correct? 2 alpha, 2 beta chains in hemoglobin, they are connected each other through this interdisulfide bridges, and this mainly cysteine. Likewise, even intra also for a particular shape or conformation, as I said, for the protein. Okay, this conformation is very important for the native structure of the protein, where you see the activity. Okay, so now you need to denature, means you need to cleave this disulfide so that it will become a long thread primary structure, that is peptide. So how you do this beta mercaptoethanol? In that way, it cleaves this cysteine to cysteine. So wherever interconnections are there, intra and inter, that will be cleaved irreversibly and made into cysteine. So there are no more interconnected. Understood? That is the role of beta mercaptoethanol. Then another important constituent of your sample buffer is glycerol. Glycerol is important because when you are loading your sample, that will go directly into the well. Okay, it will settle down, sample will settle down in the well. It acts like a density. It will become more denser. So that is the role of glycerol. So beta mercaptoethanol we discussed, SDS is there. So even sample buffer, you smell it, you always feel that SDS. It will even stuck to your clothes, everything, you know. And then glycerol, as I said, it is adding the density to the protein sample, and it can directly gets into the bottom of the well and settle down. Then there is a bromophenol blue, that's a tracking time, because of which the color, blue color is attained for the sample. Okay, so this way, the sample buffer, the first part of SDS page as we discussed, includes the components required for denaturing the protein. Understood? Any questions?
So what is the role of EDTA?
So here, apart from the components which we discussed, right, you need pH, proper pH, okay? And this pH maintenance is done by this is a buffer component. Next, we are going to discuss how the pH is important for SDS page. Okay, this is a buffer component to maintain the pH. Then,
Okay, sir.
yeah, then the second part of SDS page is gel polymerization. Gel polymerization in case of polyacrylamide gel electrophoresis is mediated by free radical reaction. Ammonium persulfate, when you add, followed by TEMET. This TEMET is the trigger for ammonium sulfate to form the sulfate free radicals. Then these sulfate free radicals responsible for formation of the acrylamide bisacrylamide meshwork. You see here in the illustration, this is the bisacrylamide And here you can see the pink color stuff. This is the kind of a cross-linking agent. These are the acrylamide monomers and interconnected through this bisacrylamide cross-linking agent. And this way, a meshwork will be formed. Unlike in agrogel electrophoresis, where I said you are heating, then allowing it to cool, then it will get solidified and it forms a gel. In polyacrylamide, you have all the components. Then finally, you are adding ammonium sulfate and temate, acrylamide, bisacrylamide, ammonium persulfate. Then finally, as soon you add temate, this temate is responsible for converting ammonium persulfate to form sulfate-free radicals. And these sulfate-free radicals are responsible for formation of polyacrylamide gel network. Gel is basically a kind of a meshwork of these monomers. What are the monomers here? Acrylamide. So acrylamide monomers cross-linked by this bisacrylamide and forms gel. And this is the solid support, and this is the basis of zone electrophoresis, that is SDS page. Understood? Okay. Next is electrophoresis. So if you see this gel, there are two distinct parts, the bottom portion and the upper portion. These two portions are separate. When the gel is having two distinct or any distinct parameters, what type of gel it is?
Gradient gel, sir.
Very good. It's a gradient gel. So here this gradient is because of the concentration and pH. So here I explained the pH in these two called as, upper one is called upper gel, that's stacking gel, and lower gel is called resolving gel. Resolving gel because actual resolution of the protein is taking place in the lower gel, and that is called resolving gel. So resolving gel and stacking gel, they are distinct. That's why it is called gradient gel electrophoresis, your SDS page. Here there is a little bit misposition of these boxes are there. I mean these boxes here to highlight this pH 6.8, that is your upper gel, and pH 8.8, that is your resolving gel, where actually proteins are getting separated. And stacking gel is for the stacking of the sample. So you are loading, these are the wells, there's a comb, and then as I said, you are mixing all the contents of required for polyacrylamide gel. As soon you as ammonium sulfate followed by your beta mercaptoethanol, immediately you have to load, and you made two of the beakers. One beaker is for separating gel, and these two beakers are different with respect to the amount of acrylamide, nothing but that form the pore size. Okay, so the pore size is very high in the stacking gel, about 5% we use, and then the resolving gel, as I said, the different concentrations we are using. These different concentrations depending on what is the size of the protein for which you are looking at. Okay, depending on that, you have to choose which concentration of acrylamide is appropriate. Okay, so now as soon you add temate, immediately you have to pour first your separating gel, then you have to pour after solidification. You can wash it, and then you add the stacking gel, and then immediately we insert the comb. Leave it for some time, then it gets solidified. So your gel is ready now. You take out the comb, and your wells you can nicely see for your sample loading. They are ready. Understood? So now these gel plates, you can fix it, and this is the way apparatus that you have arranged for your loading. And you have black and then negative electrodes. So top is black. What is black means? What is the charge?
Negative.
Negative, sir.
Negative.
And what is the charge of your protein?
Negative.
Negative.
So now where it has to move when you
Positive.
forces.
Positive.
Positive.
Towards anode.
So where is the positive electrode in this apparatus?
Anode.
And
Anode.
at the bottom.
Yeah, at the bottom. So that's why your wells are at the top portion of the gel, and you can see here this is the appearing like blue color, right? Because you have in your sample buffer bromophenol blue that has added the blue color and that is a tracking tag. You can make out where your sample front is there. Okay, so that way now you can make sure that you have completed your electrophoresis for your sample. Take this gel, and you see here already resolution of the protein has taken place, but you cannot see anything. It is clear unless you stain with some staining reagents like Kumasi, which is widely used. When you stain with Kumasi, then your gel is going to be this way. This is very good gel. I don't think it's a good gel because there is some degradation of the protein here. But you see, this is a standard marker. And now people are using this pre-stained markers. This is called kaleidoscopic markers are available. Maybe in Syngene you can see these markers when separated, as I told, you cannot make out when gel is running. But if you use the pre-stain marker, you can see when protein is running, the stains, the stain markers will appear like this. Okay. No need to go for Kumasi stain even before itself, you can make out the separation. And this is the way after Kumasi stain you can see, but usually after STS page, unless you want to go for proteins, further analysis like MS spec or, for characterization purpose, we don't use this SDS page. Routinely, this is used for Western blot analysis. And these are the different proteins in your sample. Western immunoblot technique is to analyze particular protein in this. So this is based on this immunoblot technique. Wherever the term immuno is coming, it is based on antigen-antibody interaction. Okay. So antibody here, antigen is present in the sample here, and you are using antibody for that particular antigen which you want to detect. And when you use this gel, from the gel, you are transferring the proteins to the membrane. Okay, that is called nitrocellulose membrane or PVDF membrane. And that membrane will be incubated with primary antibodies, secondary antibodies, and then chromogenic substrate again. That will give this type of immunoblot. Okay, so here in the original, you have seen all the proteins, but after immunoblot, only particular protein you can visualize. Understood the application? This way, STS page is done regularly as a basic technique in the laboratories. And here, as I said, different molecular weight ranges and gel concentration that you need to choose accordingly. What is your required protein or the protein of your interest that you are looking at? And what is the range of gel concentration that is appropriate, right? So that is all about STS page. And any questions as of now, you can ask.
So what are those numbers in pre-stain markers? Or is it molecular weight or something?
Yes. These are all kilodaltons.
Kilodaltons.
These are all kilodaltons. Okay.
Okay.
Any other questions? So applications or analysis of these samples after separation on SDS page. In this slide, I just given you with the depictions. So you have now, as we discussed, the quaternary structure, proteins with the multimeric, many monomers. For example, here, dimeric means two polypeptide chains are connected. Trimeric means the native protein with three trimeric. Okay. Tetrameric, four different monomers. Now, this is under native condition. If you separate like activity gels and gymograms, as we discussed, proteins get separated as it is with this quaternary structure. As I said, quaternary structure means like hemoglobin, 2 alpha, 2 beta together. And here what happens in SDS page, because you are boiling, I didn't tell, I think in sample buffer preparation, see here, these are all the contents I told, and you have to boil the samples. When you boil, what happens? Because of beta-mercaptoethanol and STS, this will become like a primary structure, a thread-like structure of this polypeptide, and SDS is going to bind. SDS is a negatively charged, and this detergent is going to bind to polypeptide in such a way that for every 2 amino acid, 1 SDS will be bound. Okay, so that way this whole protein will be uniquely negative charge. Understood? So that is what you are making during the sample preparation. So that's how now your quaternary structure has become monomeric structure, correct? That is what your primary structure. Now, if it is a whatever it is in this depiction, after separation, you will only observe two bands. Okay, correct? So here there is two sphericals and triangle is there. Two sphericals, you have to imagine they are of same size. As the size is same, though they are different with respect to the number, but when you do SDS, they form only one spot. Okay, so if it is a tetrameric structure, trimeric structure, you will feel after SDS page only two units are there, which are separated based on their molecular weight. Correct? Correct? Now, this is in that way, it is incomplete information in SDS page. What next to know that how many subunits for a particular protein? That is called isoelectric focusing. Okay, so you need to, so first in SDS page, the separation is based on their molecular weight. There is no effect of charge, unlike in native page, because the charge is, the native charge is made unique negative charge because of the SDS addition. Correct? All are uniquely negative charge. Now the separation is only based on the size of the protein. And as I said, based on the mass, though there may be two proteins with the same mass, you can't make out and it will appear as a single band. So to separate it further, that is the technique, two-dimensional gel electrophoresis, two dimensions, two different parameters. Earlier it was mass only the parameter in SDS page. In two dimensions, the separation is based on the first dimension and second dimension. First dimension is based on separation on the basis of isoelectric pH. You have told that amino acids are charged. So based on the native charge, whatever may be the charge, as I said, based on the acidic amino acid and basic amino acid content, the native charge of the protein depends on, and that charge is the basis for the first dimension separation, that is isoelectric focusing. It's also called as tube gel electrofocusing. Okay, then the same samples, you are continuing the experiment with the second dimension separation, and that is called the normal SDS, the denatured page, that is based on the mass. So first dimension is based on the isoelectric point or isoelectric pH, and second dimension separation is based on the mass. That way, your two proteins in SDS page in the depiction you observe two subunits with the same mass as a single spot. They appear different because they are different with respect to their isoelectric pH or isoelectric point. Now, what is isoelectric pH or isoelectric point? You must be knowing deuterion. Any chemistry student, can you tell what is deuterion?
Yes, sir. So when positive and negative charge on amino acid or any compound exit simultaneously, they are known as deuterion.
Deuterion. So, as I said, acidic or basic amino acid content is the requirement, or the one which decides the net charge of the protein, and it is variable with respect to type of protein. When you apply electric field and when you are separating on the matrices which are with a different or gradients of pH regions, separation occurs when that particular protein will become deuterion, means both positive and negative charges appear on the protein. And when it becomes neutral, further electric field will not have any impact. When up to that particular pH, up to that particular deuter, that is called isoelectric pH. When it is attained, there will be no effect of electrophoresis. Okay, and that is very, very, very precise and very, very sensitive, that two proteins which are with the same mass can have distinct isoelectric points or isoelectric pH values. This is the basis of tube gel electrophoresis, first dimension separation. Understood? Right?
Yes, sir.
So that is what now isoelectric focusing or tube gel and tube gels. So like capillaries earlier we discussed, these are bigger than the capillaries tubes. We are polymerizing the gel, and this is called urea electrophoresis, because unlike SDS, in SDS page, the sample buffer, you have urea. Why urea and SDS page, how it differs? I told that isoelectric focusing first dimension separation, you need to take care of the nativity of the protein. So protein should not be denatured. So urea in that way, it is okay. Okay, you are denaturing, you are separating it based on the molecular weight. Sorry. So you used to denature the protein, but it will not alter the intrinsic charge. Okay. The intrinsic charge will not be disturbed because of the urea. But I told that SDS, when you add, it is imparting the unique negative charge, correct? So that effect you will not see when you use urea. And that is the tube gel electrophoresis, the first dimension electrophoresis. And I also told that you are separating the first dimension sample components. So you have denatured here, urea. Ampholytes. These ampholytes in the gel forms a gradient. These are nothing but the polymers of acids and base functional groups. Acid and base polymers at different ranges, it forms particular pH regions in the gel. Now, when you run the sample, as I said, the proteins will move until that pH where it is corresponding isoelectric point is attained. Then further electrophoresis will not have any impact. Okay, so that is what ampholytes or biolites, that are the parts of your gel constituents in a tube gel. Now, applied electrophoresis leads to migration until that particular proteins attain isoelectric point. That is all about the tube gel electrophoresis. Next one is you have to carry on the first dimension separation with the second dimension separation, and second dimension separation is like your SDS page. So this is what tube gel electrophoresis that we discussed. So these gel constituents are having ampholytes. Ampholytes are polymers of acids and bases that forms. See here, they are acids and base functional groups. And when you pre-run, we do pre-run. Without sample running, when you run it, this will get arranged in a particular pattern. So these are the gradients of different pH regions in the gel. Then you load the sample, and now you do isoelectric focusing, and that results in the separation of proteins based on their isoelectric points. For example, here there are two proteins which may be having, I'm just giving example, it may be of same mass, but it will be having different isoelectric point. Okay, they may differ. So that is the basis of isoelectric focus. Understood? Any questions? So this is the first dimension separation. Our technique is not over. Two-dimensional separation. That is two parameters. First dimension is tube gel. Now the second dimension is separated. This is the for first dimension. We are using this gel apparatus. So tube gels, you have to fill the material and then you have to fit it here. You can make out. This is the old classical method, but now people use the pH strips. Directly ready-made strips they use. And here, these are the slots for tubes that you can put it. And here it is the upper chamber. And what should be the charge, upper chamber and the bottom chamber?
Upper chamber, positive, and lower chamber. Sorry, bottom chamber, positive, and upper chamber, negative.
Right, clear, everyone?
Yes, sir.
Any conflicts? Anybody is having doubts, questions for what Nima Sarah has told? Clear, right? Because here tube gels, your sample you are loading on the top portion of the tubes. Then you are going to insert in the slots, and after loading the sample, you are filling this with a buffer, and samples will move from top to bottom in the different pH regions. Again, you have to choose for your protein of interest that particular pH regions, and the proteins will move to that respective pH zones where it becomes isoelectric point, for that particular protein, and it becomes deuterion. So there will be no effect of electrophoresis. Understood? So that is the first dimension. As I said, this is the first dimension gel already you have got, and from these tubes there is a method to extract these tube gels, and these gels are placed on the, again, these flatbed gel plates, and these are notched plates, and you can put the hot agar here so that continuation with the second dimension gel is made. And when you run now electrophorese, unlike your SDS page, here you see there are so many spots. This is the way you can separate now many more proteins. Okay, this is the topic here. This concept is called resolution. So this is with a higher resolution. In SDS page, for example, for a particular sample, you have separated into 50 different proteins, and here you have 50 proteins you are observing as suppose 1000 spots. Means same sample resolution, you are increasing number of spots or number of sample spots that you can observe. Understood? And you see, unlike SDS page, here is like kind of a confucius shattered and spots. Now, very important topic here is how do you analyze this type of gels? Clear? So this two-dimensional gel electrophorese is a high-end proteomic technique, and for samples to separate it out. And why you know all of you why you need two-dimensional? As in SDS page, we have separated based on the mass, and two-dimensional separation will give you based on isoelectric point and also based on mass. So two proteins with the same mass, you can separate it out in the two-dimensional gel electrophoresis. That way, 50 spots or 50 sample components you are observing in SDS page become 1000 components, for example. So that is called resolution. You are increasing the resolution of the sample analysis. Okay, and for the analysis of two-dimensional gel electrophoresis, there is a technique called difference gel electrophoresis technique. So here, like in this illustration, you have 3 different colors. Can anybody tell what are the 3 different colors that you can note here?
Yellow, orange, and green.
Right, good. So green color, I can see yellow color and red color. Okay, this is the basis for your analysis. Now, you have sample and you are comparing your sample or control with the control versus sample. Now, what you will do, one sample and one control, use distinct fluorescent probes. So yellow color for your control, and your sample you have used green color. And when you mix and load in the tube gels, no, you have separated it in different tube gels and run it on the gel. When you see here, the colors are mixed now. Wherever the red color is there, those are the proteins expressed in both in control and samples. Okay, and the yellow colored one, they are coming from the sample. Green color one, they are coming from the control, like that, whatever it is. Understood the logic? So you have now control and sample, and you are using 2 different fluorescent colors. Control, you are using yellow color, for example, and sample with a green color. When you mix the samples and separate it by 2 dimensions, and this appears respective colors, whatever the sample and control. But the proteins which are present in both sample and control, they will appear 3rd color. Correct? That way you can easily pick, and there are softwares available to make it out automatically and analyze these spots, nothing but the protein components of the sample. Okay, so this is the way the 2-dimensional gels are being observed. That is all about SDS page technique. And next we are going ahead with immunoelectrophoresis. So immunoelectrophoresis, I told already during immunoblot analysis, wherever the word immuno is coming, those techniques relies on antigen-antibody interactions. Antigen-antibody, all of you know, they are specific, like enzyme substrate, they have got biological affinity. The same way, particular antibody is specific for particular antigen. They are very specific. Antigen-antibody is a very broad word, right? So that way, this immunoelectrophoresis relies on antigen-antibody specificity. So you need to detect, this can be used for diagnosis. So whenever in the sample, you have unknown sample. So you are suspecting one particular case. Okay, that sample you can load in the well, and this is agarose on the slide. And in another well, you know, antibody for which you want to test whether it's present in the sample or not. So if the sample is having that particular antibody, then it forms the precipitin line because of this antigen antibody complex formation. Okay, antigen antibody, they are specific and they form the complex, and that appears as a precipitin line. So appearance of the precipitin line in the gel is an indication for positive reaction. This is called immunoelectrophoresis. So you have to load in the two wells antigen, that is from the sample, and antibody for which you want to analyze. When you run the electrophoresis, precipitin line observation gives you the positivity of the sample for that particular antibody. Understood? That way it can be used for diagnosis. Then the same type of electrophoresis related to immunological technique, antigen antibody types analysis, that is called rocket electrophoresis. Rocket, you know, what is rocket? Here it is, rocket is different meaning. This is because of the shape. So rocket shapes of the precipitin lines. That's why it is called rocket electrophoresis. And in this case, here you see both antigen antibody are moving in the agarose. That's why it is called double immunodiffusion technique. Here, only antigens will move. That's why it is called single immunodiffusion technique. Your agarose, you are going to add your antibody, then polymerize, and wells you have created in which you are going to load your samples. And these samples with antigens, concentration, you have taken here standards. You see here, there is a particular pattern of rocket size or length. And that is based on the concentration of the standard that you have used. And you can use your sample in the last well. Compare with your standards amount, and you can calculate like your colorimetric enzymasis. You can correlate from the sample unknown with that of the known standards. Okay. So rocket length here, first precipitin line, as here it is a positive indication, and length of the rocket gives you the amount of antigen. So you can both qualitative and quantitative test here, right? Even you can quantify. Understood? This is called rocket electrophoresis. Any questions? So that is about the rocket immunoelectrophoresis, and there are two more techniques which are not more oftenly used, but I'm going to give you a gist of the contents for blue native gel electrophoresis is one more technique. So blue native, again the word you should appreciate native, that is for the original conformation of the protein when it is present in the living system, that is functional state of the protein. So you are non-reducing we have used, right? For native gel electrophoresis, it is non-reducing or non-denatured. So protein remains in its native state. So the first separation is non-denatured condition, and then the second separation is denatured condition. This is the technique. So this again two-dimensional technique. First separation is under non-denatured, and second dimension is under denatured condition. So this is also another technique which is used for particularly to analyze the multi-protein complexes like mitochondrial electron transport, or to characterize again how many monomers are present in the protein. How many monomers in hemoglobin? How many?
4, sir.
Right.
2 alpha and 2.
Okay.
4.
So now
2 alpha and 2 beta.
right. So what is a monomer? Now I understand that you all know that. So here multi-complex proteins with many more number of monomeric polypeptides can be analyzed in the protein based on this blue native gel electrophoresis. Okay. Now the last one, that is another important one, diagonal gel electrophoresis. So diagonal gel electrophoresis, because the term you see here, diagonal. Diagonal is the shape of the protein that you will observe in the gel. And this is also two-dimensional. First dimension is under non-reducing condition. You see here, this is the dimeric protein with two subunits. And what happens after denaturation? It forms a diagonal if the proteins are not having any cysteine bonds. I told cysteine, right? Cysteine means here in the depiction you can see. So the cysteine bonds are very important for the quaternary structure of the protein. These are responsible for the quaternary native structure. So you are having the connection within the monomers, that is intradisulfide bonds, and interdisulfide bonds are responsible for connecting different polypeptide chains in a multimeric quaternary structure, native structure of the protein. So for denaturation, you need to use, when you use SDS and beta mercaptoethanol, this is cleaving this particular bond, reducing this, and this is coming from your cysteine, right? Sulfur containing amino acid cysteine. Understood? So that way, where is this cysteine bond position? Okay. So for the confirmation of the protein or protein structural studies, this diagonal gel electrophoresis has got a very important application. To know the position of this cysteine bond, this technique is used. Now, after this first dimension under non-reducing condition and second dimension under reducing condition separation by electrophoresis is done, this diagonal indicates that these are the proteins which are not having any disulfide. And you can see maybe in the afternoon classes in Singing, you can get exposed to this technique. Certainly, they will be doing this technique and how they will analyze these spots. Okay. So that way, diagonal gel electrophoresis is another important electrophoresis technique which is used to know the position of the cysteine, okay, disulfide bonds, and that is very important for the conformation of the protein. So this is the application of the same which we discussed. So the protein analysis is very important for the technique to define which protein and what type of conformation that it is having. So that is all for the first part of our lecture. And any questions, anybody, you can raise now. This is the last chance maybe. After this, I'll continue with capillary electrophoresis. Now we'll take a break for 15 minutes, or maybe half an hour break. Okay, please come back after now 10:50 we'll raise you. Now it is 10:20. What time? 10:50 we'll raise. 10:50 we can resume with the second part of the lecture. Please stop.